Hello students, this is biology revision class. The revision topic is enzymes, and the main contents to be covered in this revision are nature of enzymes, functions of enzymes, and factors affecting enzymes. Till the end of our program, stay tuned. In today's lesson, there are three main contents that are there: the nature of enzyme, functions of enzyme, factors affecting functions of enzyme. Start with the nature of enzyme. As you know that enzymes are globular proteins. All enzymes are globular proteins. Globular proteins all have a unique tertiary structure. Show a model of tertiary structure of a human lipase enzyme in this slide. You can see on the left side. So this enzyme hydrolyzes lipids into fatty acid and glycerol. It has a region called the active site. Part of the enzyme molecule that binds with the substrate molecule, join with the substrate molecule, so that the enzyme can catalyze the chemical reaction in biological process. So the active site of enzyme shaped to follow here on the left side, the hydrolysis of sucrose by sucrase, binding with a particular substrate, and that substrate only a single enzyme is going to catalyze a single type of substrate. So binding such a way that the reaction can take place requiring less energy than if the enzyme was not present. So example here, you can see that the enzyme sucrase catalyzing the hydrolysis of sucrose into glucose and fructose. The enzyme is unaltered by the reaction. So we're going to define what an enzyme is. An enzyme is a globular protein with a uniquely shaped active site. It acts as a biological catalyst for a specific reaction, but remain unaltered by the reaction. Catalyst is a substance that speeds up a chemical reaction and remains unchanged at the end of the reaction. So you can see here there are different enzymes do have different active sites. The substrate and the active site should bind together. That should be fit to the active site, any substance. So each of these enzymes do catalyze their own substrates. So they have unique active site. Now we're going to look at the properties of enzymes. All enzymes are proteins. They are biologically catalysts. That means they speed up a chemical reaction without being used up. So they can be used over and over again. They are specific. This means they catalyze one reaction only. A small amount of enzyme can bring about a change in a larger amount of its substrate. So here you can see the catalysis process, how enzymes and substrate molecules bind together. They form enzyme substrate molecule. Then they bring change and produce a product. But the enzyme remains itself here. And enzymes are affected by pH and temperature. They do have their own optimum temperature and optimum pH to catalyze a reaction. Beyond this and below this, the activity of enzyme will be affected. So they can be destroyed by excessive heat. They are also affected by the concentration of the substrate and the presence of a certain substance that act as inhibitors. So that temperature, pH, and the presence of certain inhibitors will hamper or will affect the catalytic activity of enzyme. What are catalysts? A catalyst is a substance that speeds up a reaction. The reaction itself is unaltered. There is no overall change to the nature of the product, the energy change that takes place during the reaction, the catalyst itself. So at the end of the reaction, all these things are occurred to a catalyst. So catalysts are substances speed up a chemical reaction. The reaction itself is unaltered. Why are enzymes specific? The answer to this question is very simple. This is also a function of the active site. It is because of the active site, numerous enzymes do have their own specific active site, so that they are called as specific. Enzymes are specific because of their own unique active site. Because of the conformation of the active site, the way in which it is shaped makes enzymes specific. Only a certain substance or combination of substance can bind with it, because only one substrate or substrate combination can bind at a time. So said enzymes are specific. There is only one possible reaction that can be catalyzed due to their specificity. So here in the slide, you can see that different shapes of the active sites and different substrates. How are enzymes affected by pH and temperature? as we have said that enzymes catalyze a reaction with optimum temperature. There is a preferable temperature for different types of enzymes. So they are properly working at this optimum temperature. Beyond this and below this, they will not properly catalyze a reaction. So a higher temperature gives an enzyme molecules and their substrate molecules more kinetic energy. They move around faster and form more enzyme substrate complexes. As the temperature exceeds, increasing gradually, so it increases the kinetic energy of the substances, the substrate molecules, to bind with the active site. They move around faster and form more enzyme substrate complexes. A higher temperature affects the chemical bond holding the tertiary structure of the enzyme in a place, particularly those in active sites. As more and more of these bonds break, the shape of the active site changes, can no longer bind with the substrate. Temperature can affect in this way. Excessive increase in temperature will break the bonds of the active site particularly, so no more they can catalyze or no more they can bind with their substrate molecules. This way they can affect the catalytic activity of the enzymes. When we come to pH affects the enzyme molecules also in two ways it can affect. Likewise, temperature affects in two ways. A pH that is too low, too acidic in other words, and a pH that is too high, it means too alkaline, will cause changes on the active site to alter and cause active site to lose its conformation. The substrate cannot bind and so the reaction no longer catalyzed. So this way a pH can affect the enzyme activity. So enzymes do have different names, and how naming and classifying enzymes have been started, and we're going to look with specific examples. First, how they have been classified or grouped into certain categories. Let's look at some of the examples of enzymes, like lipase, for instance. This is catalyzing or hydrolysis of lipids. ATPase, this enzyme is involved in the hydrolysis of ATP molecule. Succinate dehydrogenase, these are involved in the removal of hydrogen ions from succinate during respiration. DNA polymerase, this enzyme involved in the joining of nucleotides to form DNA. Pepsin, digestion of proteins in the stomachs of mammals. So these are some of the examples how they are involved in a certain reaction, but how the naming is carried out in the spheres of the enzyme. Different enzymes are named in different ways. Most common enzymes are named by adding AS. So this is arbitrary naming of enzymes. AS to part of the name of the substrate. By adding AS, enzymes can be named. Like example, lipase, lipid hydrolyzing enzyme that we have mentioned earlier. Sucrase, this enzyme is a sucrose hydrolyzing enzyme. So the substrate has a suffix AS. Enzymes are named on the basis of the reaction they catalyze also. Like polymerase, aids in polymerization, joining of similar unit together. Dehydrogenase, removal of hydrogen atoms or ions. You can see in this slide there are different names of enzyme, their sorts, and how they can be applied in different process of biological activities. So different enzymes are named in different way, we have said. So still we have different mechanisms or different namings here. Some enzymes have been named based on the surface from which they were first identified. For example, papain from papaya. Others are named according to where they act. For instance, intestinal protease on the intestine. Some enzymes end with IN, indicating that they are basically proteins. Example, pepsin, trypsin. Usually have alternative name that tell you rather than more about them. Example, the alternative name for pepsin is gastroprotease. So the intestinal enzymes they have alternative names, like the pepsin do have gastric protease. So a number of proteins enzymes are there in the gastric or in the process of food digestion in the living organisms of human and other animals. So because of ways in which enzymes have been named, biologists at the Enzyme Commission decide to produce a systematic way of naming enzymes based on the way in which the enzymes act. So this arbitrary naming that we have mentioned earlier are not being used in this modern era, simply to just how the process of naming enzymes been started in the past. So today in the modern society, enzymes have been named in a systematic way. There is a commission, the Enzyme Commission decide to name enzymes based on the process they catalyze. So accordingly, the classification and the systematic nomenclature of enzymes have been come into practice. So on the basis of the type of reaction they catalyze, enzymes commission naming those different enzymes. So accordingly, 6 group of enzymes can be recognized on these bases. These are on this slide: oxidoreductase, transferase, the third hydrolyses, the fourth isomerase, the fifth liases, the 6 ligases. All these are 6 classification of enzymes are there. All enzymes in this world are classified according to appear in this order, okay? And also they do have suborders under these main classifications. Under these subclasses also there are subclasses. So each of class of the enzymes contains several different but related subclasses. Each subclass is further divided into subclasses. Within the subclasses, each enzymes has a number. So you can see here in this slide, for instance, the enzyme EC2 means here transferase. The first EC1 is oxidoreductase. So EC2 do have 9 subclasses. Under these 9 subclasses, there are subclasses, 2.91. You can see in this slide. And under these subclasses, there is a number. 1 is at the last. So 4 digits are there in this EC2 commission of classification of enzymes, that is a transferase. So here an enzyme named N-senyl transferase selenium transferase. This is the name of the enzyme according to the enzyme commission. So here, so in the systematic naming of enzyme, an enzyme have a name such as EC 3.4.11.1. Each part of the description tells us something about the enzyme. So here, each of the enzymes that we have mentioned, each and every section of the division goes like that. So here, enzyme EC 3.4.11.1. Each of these number refers, the first 3 refers to the enzyme is hydrolase. The second 4 number refers to a peptidase. 11, the third digit 11 refers to aminopeptidase. And the last digit number 1 refers to leucyl aminopeptidase. So this way, the enzyme commission start to name those 6 classes of enzymes with their subclasses, subclasses, and numbers. So you can see here in the left side of the slide how different enzymes have been named. Let's look at some of the use of enzymes. Enzymes can be used in different areas, in different sectors. For instance, in the dairy sector, in detergent sector, in textile sector, in food processing sector, in pulp and paper sector, medicine, pharmaceuticals. So in all these areas, enzymes do have various tasks. So our day-to-day life is not separated from enzyme activity. So this table tells you that a lot of functions of the enzymes, leave alone that the injera, the bread, milk that we are using in our day-to-day lives are not separated from enzymatic activities. So different industries like the beverage industries, the brewery industries, they involve enzymes for make easing the process of life. Enzymes are also involved in carbon reduction, okay? Enzymes in carbon dioxide emission reductions. You can see that these are on this slide. Different industries are involved, like the laser industry, the pulp industry, the cheese and the bread industries. All of these do have a certain amount of carbon dioxide to be emitted from per ton of or from 1000 kg or from 1 ton of process of the laser or pulp or cheese, whatever. So each of these process, when they have these enzymes, they reduce carbon emission to the atmosphere. By this way, they are involved in reducing carbon dioxide to not to be exist in the atmosphere, so that they will contribute for minimizing the global warming. So using enzymes in industry can help to the environment, because enzymes allow some manufacturing process be carried out at lower temperature. Less carbon dioxide is produced, in raising the temperature of the reactions. The next figure here show you the mass of carbon dioxide emissions saved in some process that now use isolated enzymes rather than traditional method. So each of these do have their own emissions. Now it is time to see some revision questions. The first question: Identify the enzymes that are used to cut DNA molecule into small pieces. The alternatives are A. Urases, B. DNA polymerase, C. Reverse transcriptase, D. Restriction indole nucleases. So the answer to this question is D. Restriction indole nucleases. So that DNA into pieces, process of into small pieces that used in chemical process. Second question, which part of the human alimentary canal contains digestive enzymes that function at acidic pH? A. Stomach, B. Mouth, C. Esophagus, D. Small intestine. Answer to this question is A. Stomach. Number 3. Which of the following is made of globular proteins? A. Enzymes, B. Collagen, C. Keratin, D. Glycogen. Answered A. You are correct. That is enzyme. Question number 4. Which of the following term refers to how fast an enzyme acts on its substrate? A. Turnover, B. Enzyme number, C. Substrate number, D. Product number. The answer to this question is A. Turnover. If you say A, you're correct. The last question in this part of the lesson, question number 5. Which class of enzyme do the digestive enzymes belong? A. Sterases, B. Hydrolases, C. Transferases, D. Isomerases. If your answer is B. Hydrolases, you're correct. Now, let's go to the other part of the enzyme section. This is the second section, that is functions of enzymes. Now, in this section, we are going to look at the different functions of enzymes. So how do enzymes act as catalysts? Here, catalysts, as we have mentioned earlier, speed up chemical reactions. In order for molecules to react, they must have sufficient energy. The energy to start off the chemical reaction is called activation energy. For short, it is written as EA, activation energy. Imagine a reaction in which substance A reacts with substance B to form substance AB. We can write this as A plus B equals to AB. So for a reaction to begin, there must be a sufficient energy. This energy is supplied from the activation energy in the process. So here in the graph, you can see that uncatalyzed reaction and catalyzed reactions. The red line indicates catalyzed reaction. The blue line show you uncatalyzed reaction. So uncatalyzed reactions will consume a lot of activation energy. The catalyzed reactions are using a little activation energy. Apart from this, within this catalytic process, the enzyme can catalyze the reaction in a number of times, again and again for the process. In a short period of time, they can produce the required product very quickly. But uncatalyzed reaction goes on slowly. However, it produces the same product. The product is not as such sufficient enough. Slowly it producing, time consuming, energy consuming a lot, but the product is little. So that an enzyme catalyzed reaction gives you a product sufficiently with less energy. The enzymes can be used several times repeatedly. So the equation gives only the reaction. Here A plus B equals to AB, as we have mentioned, as I mentioned earlier. So it only the equation gives you only the reactant, starting material, and the products. It does not show how energy changes as the reaction takes place. The reaction must climb an activation energy hill, as I mentioned earlier, before anything happens. Under normal condition, very few molecules of A and B have sufficient kinetic energy to climb the activation energy hill. So the reaction proceeds slowly. A catalyst lowers the activation energy required for the reaction. So that, as I mentioned earlier, the activation energy is very important for the enzyme to start off. So more reactants molecules can meet this lower energy requirement, and so the reaction proceeds more quickly in the inclusion of enzymes. Because the enzyme molecules is unaltered by the reaction, it can be used over and over, and so a small amount of enzyme can affect a large number of substrate. So this way the enzymes are catalyzing a reaction. How do enzymes lower activation energy? The answer to this question is, before we answer to this question, let's look at, there are two models of enzyme actions. The first one is the lock and key model, first proposed in 1894 by the German biochemist named Fischer. You can see here in the black photograph over here is a Fischer who proposed the lock and key model. The second, the induced fit model, proposed in 1958 by Koshland. You can see the Koshland here in a colored photograph. Both of these models suggest that the enzyme catalyzes the reaction by lowering the activation energy. Both of whether it is the lock and key model or induced fit model, their main activity is by lowering the activation energy, they can produce or they result a product in a short period of time. In particular, they differ in explaining how the substrate binds to the active site of the enzyme. The lock and key model differ from the induced fit model. How do they bind with their active site? In this way, they differ. In lock and key model, the enzyme is complementary to the active site. In the induced fit model, the enzyme adjusts or reshape itself to bind to the substrate molecule. So this is a discrepancy, a difference between the two models. The lock and key model, as you can see here as a key and lock on the left side, the model is proposed that the shape of the substrate molecule are complementary to that of the active site, rather like the shape of a key is complementary to that of a lock of it fits. Okay, so like a key and lock, the name lock and key model proposed. Example, here you can see an egg sitting in an egg cup also is an example for lock and key model. The complementary substance molecules binds with the active site of the enzyme to form enzyme substrate complex. When a substrate comes, join with the active site of an enzyme, this time it forms enzyme substrate complex. The complex causes the reaction to enter transition state, in which the activation energy of the reaction is lowered. So when they enter to the transition state, the activation energy is lowered. The reaction takes place and the product formed are released. The lock and key model of enzyme action suggests that the enzyme lowers the activation energy by providing an alternative path for the reaction. Here are the alternative paths. Reactant A plus reactant B gives you product AB. This is one way. Another way is enzyme catalyzed pathway. The first one is the non-catalyzed pathway. So here the lock and key models, you give you two paths. One is the non-catalyzed pathway, A plus B, reactant A plus reactant B, it gives you product AB. In enzyme catalyzed pathway, reactant A plus reactant B plus enzyme gives you intermediate plus product AB plus enzyme. So this is two alternative ways that mentioned earlier. This model seems the enzyme substrate complex as an intermediate, which is part of a pathway that requires less energy than the normal pathway. However, a weakness of this model is that it does not explain how the intermediate reduces the activation energy. So this is the drawback of this model, the lock and key model. You can see here in the left side, in the right side of this slide, you can see how the substrate, the enzyme, and enzyme substrate complex, and finally product is produced. The second one is the induced fit model. This model suggests that the active site and the substrate aren't naturally complementary in shape, unlike that of the lock and key model. These are not complementary, that is the active site and the substrate. But the binding of the substrate molecule produces a conformational change in the shape in the active site. As soon as when the substrate binds with the active site of the induced fit model, the enzyme make adjustment rather to fit to the substrate molecule. So it try to adjust itself, make accordingly the substrate molecule. So the conformational change also puts the substrate molecule under tension. So they enter a transition state and are able to react because of the lower activation energy. And in the transition state, bonds in the reactant are put under strain and break more easily. Rejoin with the other bonds to form the products. The product formed leave the active site, so the enzyme utilized again. Most biologists now prefer the induced fit model over the lock and key model. Explains other properties of enzymes, such as enzyme inhibition, in a more complete manner than lock and key model. So this is discrepancy or the differences between the induced fit model and the lock and key model. So the rate of the other point in this discussion is the rate of chemical reaction. Each of chemical reaction do have their own rate. So we will see with these certain examples. The rate of chemical reaction is the rate at which reactants are converted into products. This is what we call it, the rate of chemical reaction. The rate at which the reactant converted into products. That is the rate of chemical reaction in an enzyme control reaction. This is determined by how many molecules of substrate bind with enzyme molecule to form enzyme substrate complexes. The number of molecules of reactant that form enzyme substrate complex with each molecule of an enzyme per second is turnover rate of the enzyme. So each enzyme do have their own capacity to catalyze their own substrate, varying in a per second. In 1 second, what is the capacity and the ability of each of these enzymes? How many substrate molecules can be catalyzed? In this left side, you can see that in the table, rate of chemical reactions are the rate of enhancement and given, and on one side the enzyme is there. For instance, OMP here, orotidiene monophosphate decarboxylase. This enzyme catalyzes a reaction 1.4 times 10 raised to 17 substrate molecules per second. This is very huge capacity of this enzyme. So the staphylococcal nucleases, this enzyme catalyzes in per second 5.6 times 10 raised to 14 times substrate molecules catalyzed. So this is a huge molecule. So this is what you call it, the turnover rate of each of the enzyme. And furthermore, you can see the remaining part of this table. And when we proceed to the other point, why do some enzymes need cofactor? There are some enzymes that need chemical molecules to carry out their catalytic activity. Sometimes an active enzyme isn't just a single molecule, but it is made of from two molecules, neither of which has enzymatic activity without the other. The two parts are the apoenzyme and the cofactor. You can see that in this right side of the slide, the apoenzyme is there, the cofactor, and they bind together, then they can catalyze a reaction. Without this apoenzyme, the apoenzyme, the enzyme part of the protein part of the enzyme, the cofactors are the non-protein part of the enzyme. These together come up, they catalyze. One without the other cannot stand or carry out catalytic activity of enzyme. So this is some properties of the enzyme that they can, some enzymes, they can use some other chemical molecules to carry out the process or catalytic process. Apoenzyme, a protein that combines with a cofactor to form an active enzyme. The protein is inactive on its own. Without the cofactor, the protein apoenzyme is inactive. Cofactor, a small non-protein particle essential for the activity of some enzymes. The cofactor combines with the apoenzyme to produce an active enzyme. When an active enzyme molecule comprises an apoenzyme and a cofactor, the whole is sometimes referred to as holoenzyme. So some enzymes carry out with this mechanism. What is inside cofactors? The cofactors include coenzymes and mineral ions. So coenzymes are organic molecules and many are derived from vitamins. Here you can see the nicotinamide dinucleotide NAD is a coenzyme. The source is niacin, vitamin. It is obtained from vitamin. So it will catalyze an enzyme, oxidoreductase in respiration. The coenzyme, they bind with an enzyme to give catalytic activity, as we have mentioned earlier. Some enzymes can only function in the presence of certain mineral ions. They bind loosely with the enzyme to give it its catalytic activity. Now take a pause for some revision questions here. Are you ready to answer the questions? Pay attention. Number 1. The low pH in which pepsin works is considered as A. Nuclear medium, B. Synthetic medium, C. Acidic medium, D. Alkaline medium. If your answer is C, correct. C. Acidic medium. Second question. The model proposed by Daniel Koshland is A. Anabolism fit model, B. Catabolism fit model, C. Induced fit model, D. Reduced fit model. If your answer is C, induced fit model, you're correct. Third question. The loss of the globular structure of enzyme is also known as A, insaturation of enzyme, B, denaturation of enzyme, C, maturation of enzyme, D, saturation of enzyme. If your answer is B, denaturation of enzyme, you're correct. Question number 4. The conversion of some substrate into different molecules in the process of enzyme are called A, catalysts, B, byproducts, C, products, D, covalent solutes. If your answer is C, products, you're correct. Question number 5. The minimum energy required for starting a reaction is called A, enzymatic energy, B, catalysis energy, C, solvent energy, D, activation energy. If your answer is D, activation energy, you're correct. Now let's go to the last part of enzyme, that is factors affecting the functions of enzyme. Enzymes are, as many other chemical processes, they do have many factors to affect them, their process, catalytic process. The turnover rate and therefore the activity of enzyme are influenced by the number of external factors, including temperature, pH, substrate concentration, the presence of inhibitors. All these factors we're going to look at step by step, and they are the major factors that are hampering or that are impeding the functions of the catalytic activities of enzymes. Must it be in the process of enzyme, as we have mentioned earlier at the beginning of this lesson, but regarding temperature. So when the temperature is raised, the particles are given more kinetic energy. It has two main effects. Free particles move around more quickly. This increases the probability that a substrate particle will collide with an enzyme molecule. Second, particles within a molecule vibrate more energetically. This puts strain on the bonds that hold the atoms in place. So enzymes working at their optimum temperature, there is a very required temperature for enzymes. They can make catalytic activity easily. So below that and above that, they will not function properly. Sometimes a conformational change will occur because of excessive temperature. Bonds begin to break. In case of an enzyme, the shape of the molecule and the active site in particular begin to change because of the high temperature. The enzyme begin to lose its tertiary structure. Remember, it is a protein and denature. As you can see here on the right side, the normal shape of the enzyme, finally with excessive heat, it became denatured, lose its natural property and shape, conformation, so that when they lose the natural shape and properties, they will not never catalyze chemical processes. So denature is the alteration of the tertiary structure of a protein. In living cells, this is reversible, a return back again. The activity of an enzyme at a given temperature is a balance between these two effects. That is, at a certain period of time, the temperature increases, the kinetic energy of the molecule increases, so that this helps us to bind with the active site of the molecule. But beyond this limit, when excessive temperature is there, so the enzymes is a protein, it loses natural property, natural conformation of the active site particularly, so that it loses the catalytic activity. No more catalysis will occur. If the raised temperature results in little denaturation, but a greatly increased number of collision, the activity of the enzyme will increase. If the higher temperature causes significant denaturation, then despite the extra collisions, the activity of the enzyme will probably decrease. The temperature at which the two effects just balance each other is the optimum temperature for that enzyme. So any further increase in temperature will cause increased denaturation that will outweigh the effect of the extra collisions. A decrease in temperature means that fewer collisions will occur. Note that the graph is not symmetrical here. Above the optimum temperature, the enzyme denatures very quickly to the point at which the shape of the active site has changed so much that an enzyme substrate complex cannot form. At this point, the reaction rate is 0. B. Acid also is another factor for affecting the activity of enzyme. The pH scale is a measure of hydrogen ion concentration of a solution or other liquid system. The pH scale ranges from 0 to 14. Solutions with a pH of less than 7 are acidic. Those with a pH of more than 7 are alkaline. A solution with a pH of exactly 7 is a neutral. The majority of enzymes in most mammals function most effectively with pH range from 6 to 8. Although the optimum pH of pepsin, an enzyme found in the stomach, is between pH 1 and pH 3. Significant change in pH affects enzyme molecule by breaking the ionic bond that hold the tertiary structure in place. This leads denaturation of the enzyme molecule like similar way like temperature, altering change on some of the amino acids that form the active site. This makes it more difficult to substrate molecules to bind. These effects occur if pH becomes either more acidic or more alkaline. You can see the graph here in the right side, the pH, the optimum pH is there. Mostly, many of the enzymes are neutral. In neutral pH, they can work. There are exceptions are there, like pepsin, which make catalytic activity between 1 and 3 pH values. The optimum pH for most of the enzymes are 7, neutral. Does the concentration of the substrate matter? Of course, the substrate concentration matters in the catalytic process of enzymes. The activity of enzymes depend on the number of substrate molecules per second that bind to form enzyme substrate complexes. If there are deficiencies of substrate molecules and there are excess enzyme, then there are little products and most of the enzymes are not occupied by the substrate molecule. So the number of substrate molecules present must have an effect. A small number of substrate molecules means fewer collisions, and so only a few enzyme substrate complex form. Increasing the concentration of the substrate means more collisions and more enzyme substrate complexes. So the overall rate of reaction is increased. Eventually, because of the high substrate concentration, each enzyme molecule could be working at maximum turnover. So the substrate concentration matters the catalytic activity of the enzyme. Where there are sufficient substrate molecules, then sufficient products, and the catalytic activity of the enzymes is at its maximum turnover. That is, each active site is binding with substrate molecule all the time, and there is no space capacity in the system. Increasing substrate concentration beyond this point will have no effect on the activity of the enzyme because all the active sites are occupied all the time. So beyond the available enzymes, the process contains more substrate molecules. This does not have any effect on the enzyme, as far as all the enzymes are occupied by substrate molecules. How much enzyme should there be? Assuming a constant large supply of substrate molecule, each enzyme molecule will work at maximum turnover. Therefore, reaction rate will be directly proportional to the number of enzyme molecules, the concentration of the enzyme. Increasing concentration will increase the reaction rate. However, increasing the concentration of enzyme will not increase the activity of enzyme. Okay, that means beyond excess enzymes are there in a certain chemical reaction, this will not increase the activity of enzymes unless there are sufficient substrate molecules along with this increment. Each enzyme molecule will be working at maximum turnover, so the activity of the enzyme is likely to remain constant. How do other substances affect enzyme activity? So there are some inhibitors in the enzyme process, are substances that bind to the enzyme and prevent them from forming enzyme substrate complex, and as a result slow down the reaction. There are two main types of inhibitors: reversible inhibitors and irreversible inhibitors. Irreversible inhibitors bind strongly to enzymes, usually by covalent bond, permanently altering the structure of an enzyme molecule, inactivating it. Here you can see on this slide, there is non-competitive inhibitors are binding with on the side of the enzyme In between is the substrate, the green one, so that the enzyme activity not further to enhance. So permanently altering the structure of the enzyme molecule and inactivating it. Example here is a painkiller. Aspirin is an example of irreversible inhibitor. It binds with the enzyme cyclooxidase 2, which is an important enzyme in producing prostaglandins, which give sensation of pain. So the second one is the reversible inhibitor. Binds to enzyme only weakly, and the bond that holds them breaks easily, releasing inhibitor. This allows enzyme to become active again. There are two main kinds of reversible inhibitors: competitive inhibitors and non-competitive inhibitors. The competitive inhibitors have molecules that shapes that are complementary to all or part of the active site of an enzyme. There is a substrate molecule, there is a competitive inhibitor. They are competing for the active site, so that by chance the competitive inhibitor may occupy the active site, so temporarily hampers or blocks the catalytic process of the enzyme. They are often similar in shape to the substrate molecules. These are the inhibitors, the competitive inhibitors. They can bind with the active site and prevent substrate molecules from binding. The binding is only temporary, and the competitive inhibitor is quickly released. A competitive inhibitor blocks the active site, so the substrate molecule cannot bind. The overall effect of the rate of reaction depends on the relative concentration of the substrate and inhibitor molecules. Each molecule of competitive inhibitor can inhibit one enzyme molecule, but only if it can collide with the enzyme molecule and binds with the active site. To do this, it must compete with the substrate molecule for the active site. Hence, the name competitive inhibitor is given. If there were 99 substrate molecules for every inhibitor molecule, then 99% of the collision would be between enzyme and substrate, and the reaction would proceed at 90% of the maximum rate. The ratio were 90 substrate molecules to 10 inhibitor molecules, there would be 10% of inhibition, and the reaction rate would fall to 90% of maximum. The painkiller ibuprofen acts as a competitive inhibitor of the enzyme cyclooxidase 2, competing with the precursors of prostaglandin, which are the substrate of cyclooxidase 2. The metabolic poison cyanide acts as a competitive inhibitor of enzyme cytochrome oxidase, an important enzyme in the release of energy in respiration. The second one is the non-competitive inhibitor. A non-competitive inhibitor do not compete for the active site. Instead, they bind to another part of enzyme called the allosteric site. This produces a conformational change in the part of an enzyme molecule that includes the active site. Because of this, the active site is a different shape and can no longer bind with the substrate to catalyze the reaction. The effectiveness of a non-competitive inhibitor is in no way affected by the concentration of substrate. Suppose there are enough inhibitor molecules to bind with the allosteric site of 80% of the enzyme molecules. 80% of the enzyme molecules will be inhibited, irrespective of the number of substrate molecules, and the reaction rate will drop to 20% of maximum. Non-competitive inhibitors are particularly important in regulating metabolic pathway in cells. How do the inhibitors control enzyme activity in living cells? So these are given by some examples over here. Many substances are produced in cells as a result of metabolic pathway. Series of reactions, which can be represented as change substance A into substance B, enzyme 1 is required. From substance B to substance C, enzyme 2 is required. From substance C to substance D, enzyme 3 is required. So E1, E2, and E3 are enzyme catalyzing reaction. All reactions in this sequence are enzyme controlled. Therefore, inhibition of any of these enzymes will interrupt the process. However, the main function of the pathway is to produce substance D for use by the cell. If the requirement for substance D in the cell decreases, then the concentration of D will increase. This is at least inefficient and may be potentially harmful because high concentration should be toxic. Such reaction sequences are often controlled by end product inhibition. End product D inhibits the enzyme controlling the first stage of the reaction sequence as shown in the figure. that is substrate A converted to substrate B by enzyme E1. So by controlling this enzyme, the end product will be inhibited, not to further catalyze and produce substance D. So this way, substance D normally acts as a non-competitive inhibitor, which prevents enzyme E1 from catalyzing the reaction that converts A to B. As a result, the entire reaction sequence is halted. When the inhibition is removed, metabolic pathway can start again. Sometimes a decrease in the concentration of D is insufficient to remove the inhibition. In other cases, the inhibitor must be removed by a substance known as an activator. Enzymes are often kept inactive in a cell as inhibitor and are activator only when they are required. So you can understand from the figure here in the right side of the slide, an example of the metabolic pathway controlled by end products, how it is controlled by previous explanations. Dear students, today we have revised topic on enzymes from grade 11 biology. I hope you find it useful. Till our next revision, goodbye.